We will continue with the gene transfer techniques. So, techniques used for gene transfer. Now, once the DNA is loaded in the vector, this DNA is again which carry the gene of interest. The next step is to introduce it into the suitable host by using certain techniques. Now, we must also understand what are the features of this host. It should be able to do replication of vector recombinant DNA, that is chimeric DNA. So once we introduce the chimeric DNA, that is the DNA of host plus the foreign DNA, then the host should be able to multiply it and number of copies should be formed. Okay, so this is what is known as gene cloning. So this is one of the essential steps. The host should be non-pathogenic, harmless and easy for cultivation. If you require the cultivation at large scale, then it should be able to replicate well. Commonly used host is E. coli that we are using in all the rDNA technology. Now let's talk about the techniques. Now these techniques are used to efficient and stable introduction of rDNA into the genome of target cell. Now these techniques are transformation, electroporation, microinjection and gene gun or biolistic method. First we will discuss the transformation. Now, transformation is the procedure by which a segment of foreign recombinant DNA is introduced in the genome of host bacterium and to increase the permeability of host cell, because it may happen that we are introducing the vector DNA into the host, but host is not taking up that DNA. So we have to increase the permeability of host when we are doing the transformation artificially. So it is the host is treated with calcium chloride at low temperature, so it is now become ready to take up this foreign DNA, or you can say vector or chimeric DNA. Next method is electroporation. Usually this is used in case of plant cell. The protoplast of host cell is created. Protoplast means you just remove the cell wall of that particular cell, it become protoplast. Okay, is created usually in case of plant cell, and the protoplast are subjected to high voltage electric pulse to create temporary pores in the cell membrane through which the rDNA enters. So under the electric field, electroporation is done. Next is the microinjection. The protoplast are immobilized on agarose and micropipettes, okay, are used to introduce the DNA into protoplast. Another method is biolistic. We also say this is gene gun method. So in this what happens, the rDNA is coated with gold or tungsten. The gold impart the stability because gold will not react with DNA, tungsten and gold, of about 1 to 2 micrometer in size. So after coating, this size particles are formed and then they are bombarded by using the gene gun in the host cell. Okay, especially used for the plant cell where the cell wall is present. So these different methods are used which are used in different you can say situation to introduce the DNA. Now next is the selection of recombinant. So what we have done, we have loaded our DNA on vector and then this DNA is introduced into the host cell by using different techniques. Now we have to check whether the host has taken up this DNA or it is not taking up this DNA. When it is taking up the DNA, then we will say that the rDNA is introduced in the host and it become recombinant. If this DNA is not taken up by the host, we say it is non-recombinant. So we have to select because there are number of E. coli cells which are present when we are doing the experiment. They are not working with the one E. coli. We are working with thousands of E. coli. So we have to select between which E. coli has insert and which E. coli does not have insert. Okay, between the transformant and non-transformant, between the recombinant and non-recombinant. Okay, so this method is called insertional inactivation. The host cell may not take up the foreign DNA. All the vectors are designed to impart the selection between the recombinant and non-recombinant. So we'll do first study in PBR322. Now this has two restriction endonucleoside GATCC, which lies in tetracycline region, and another one lies in the ampicillin region. If the insertion is made in tetracycline region, then tetracycline resistance gene will become non-functional, but ampicillin resistance gene will be active. So E. coli will survive in ampicillin containing medium, but they will not survive in tetracycline medium because tetracycline resistance gene is non-functional now. Okay, let's understand here in the diagram. This is PBR322. So it has ampicillin and tetracycline resistance. So here I have added um, two PBR322. I have added hint 3. Now this hint 3 target site lies in tetracycline resistance region gene, and foreign DNA is added. Okay, so there will be a breakage at hint 3 and foreign DNA is inserted over here. After this, we'll do the plating, that is on the medium containing tetracycline. The E. coli cells are plated, and on the medium containing ampicillin, the E. coli cells are plated. The recombinant will not grow on tetracycline, whereas they will grow on ampicillin. So recombinant are selected from the ampicillin containing medium, and here the ampicillin imparts the selectable marker. Next is we will see the blue and white selection. Okay, in blue and white selection, the cloning sites are present in Lexi gene. The recombinants are selected on the basis of color. When the foreign gene is inserted within restriction endonuclease site that lies in Lexi region, the Lexi become non-functional. The Lexi code for beta galactosidase. When the medium contains substrate of beta galactosidase, that is X-gal is the substrate of, which is a artificially made chromogenic substrate. So it produces blue color colonies of host, whereas the recombinant will produce white color colonies. So the blue color are distinguished from the white color. Uh, let's understand certain things to understand this in a better way. The E. coli colonies are round. So when you grow on the agar plate, Petri plates are there. In that we grow. We put the agar medium and then we adjust the pH. Nutrient are added, and it is solid medium. So you can see E. coli colonies round in shape. They are growing on the agar medium. Okay, so if you add any antibiotics such as ampicillin or tetracycline in the above medium, in this medium on which E. coli is growing, then those E. coli cells which have tetracycline resistance and ampicillin resistance gene in their plasmid, they are able to grow on such medium. Okay, and those E. coli cells which do not contain these genes, they become sensitive to these antibiotic because resistance is not there, no gene is there. So the E. coli cell will not grow in that case. Okay, if you add X-gal, which is a substrate, now this is known as 5-bromo-4-chloro-3 indolyl beta galactoparinocyte is what is X-gal. Substrate to E. coli medium, the E. coli cells which contain beta galactoside gene, that is LAGZ gene, in their rDNA in the intact form, okay, will form colonies, blue colonies. Okay, if the gene is intact, if it has not taken any insert foreign DNA, the gene is intact, and the beta galactoside enzyme will be synthesized. It is going to act on the substrate xcal, and since it is blue in color, so it is going to produce the blue colony. But if there is a foreign gene which is inserted, and if the rDNA has taken up this gene, then this lex z gene will become non-functional, and then it is unable to code for beta galactosidase, so the white colonies are formed. So you can see lex z gene is there. It has multiple cloning site. I have added here hind3 enzyme and foreign gene. So this is inserted. So once inserted, this lex z gene become non-functional, and it form the white colonies on agar. The E. coli cells which have not taken up the foreign DNA, they will form the white. So you can select between blue and white. So this is called blue and white selection. So this is all about the gene transfer method. Thank you.